Now we're going to do a very interesting enzyme, which is the ribonucleotide reductase. And ribonucleotide obviously is referring to this molecule in which we have the base, the nitrogenous base, and we have the sugar and we have the phosphate, right? And the thing is that obviously it's ribo, so the sugar is ribose, obviously. And what about the phosphate? Because when we're talking about the nucleotides, there can be 1 phosphate, 2 phosphate, or 3 phosphate. And the thing is that this enzyme is actually specific to what we know as the ribonucleoside diphosphate. So the more accurate name is written over here. And the name ribonucleoside diphosphate reductase clearly states the substrate specificity of this enzyme. The fact that its substrate will always be ribonucleoside diphosphate. And what is the function of this? We need ribonucleoside diphosphate reductase in order to form the deoxy form of the nucleosides, so that they can be incorporated in DNA. And the reason for this is that the only difference between ribonucleotides and deoxyribonucleosides and their counterpart deoxyribonucleosides is a difference in that in the ribose sugar of the ribonucleotide we have an OH at the second carbon, and in the deoxy we have a hydrogen. So there is simple reduction. So I'm hoping this kind of clarifies the chemistry aspect of it. So the thing is that this is going to be both for the purines and for the pyrimidines. So when you're doing pyrimidine synthesis, you will also encounter the very same enzyme. So this is a very important point because they ask you about the properties of this enzyme. First of all, for the first mark, you need to clarify the substrate. In the second point, you have to elaborate on the substrate, both for purines and pyrimidines, and what are the possible substrates: ADP, GDP, these are the purines, the purine nucleotides, and these are the pyrimidine nucleotides, the nucleoside diphosphates to be exact, CDP and UDP. And these all are converted into these counterparts. So this is the function. Now what is needed for all of this to happen? The second point to know is that the enzyme has 2 components, an R1 subunit and an R2 subunit, right? And the R1 subunit, which is this one over here, has these 2 particular bridges, or my apologies, sulfhydryl groups. And in the course of the reduction, these 2 sulfhydryl groups are oxidized. These are oxidized to disulfide bridges, and this is what allows the reaction to occur. But for this reaction to be repeated, this reaction needs to take place in reversal. And for that we have the protein coenzyme. This is another important property. They ask you about the protein coenzyme of ribonucleoside diphosphate reductase. So what is the protein nucleoside? And the answer is the thioredoxin. The thioredoxin similarly has 2 cysteine residues. And because of this, the thioredoxin, since residues sulfhydryl group is similarly oxidized to the disulfide bridge, and in order to regenerate the coenzyme in its original form, you need an enzyme. And this enzyme is very important. It is a selenoprotein which you have encountered in first year. And you also need a reducing equivalent that is donated by not NADH, not FADH, by NADPH. So this is very important. In COT, there is this really interesting schematic diagram which comes in handy if they ask you to write a note on this enzyme, and they've kind of drawn it in this manner. Technically the 2 halves need to be symmetrical, but I am bad at drawing, as you can see. Whatever. Anyways, so the interesting thing is that in addition to the allosteric sites, there will be two allosteric sites in addition to the catalytic sites. And the first allosteric site is known as the activity site, and the second one is known as the substrate specificity site, because this site is going to be governing the substrate specificity of the enzyme at any point in time. And these both are found on the R1 subunits. And in lipin cord, it's shown over here. The activity is shown by using ribonucleoside, diphosphate is converted into deoxyribonucleoside diphosphate. So that is a clear representation, one that you will find is hard to forget of what is the function of this particular enzyme. Then talking about the details of the activity site and the substrate specificity site. So catalytic site to substrate to allosteric sites. In the case of the allosteric activity site, DATP and just DATP, the deoxy form, is going to be an inhibitor and ATP is going to be a promoter. So when there is a lot of deoxyadenosine triphosphate, you have to remember the form, the triphosphate, there will be inhibition of the overall catalytic activity. And so none of these, there won't be any DNA synthesis, because there will not be the formation of DATP, DGDP, right? There will not be the formation of any of this, DGDP, DCDP or DUDP. And because there will not be any formation of this, and because it is this form that is going to be integrated, there will not be any DNA synthesis if there is a lot of DATP accumulating for whatever reason. Prevents the reduction of any of the 4 nucleoside diphosphates. There is DATP toxicity in the disorder adenosine deaminase deficiency, which is a disorder of the purine degradation. Then there is a substrate specificity site. For example, if there is a lot of deoxythymidine triphosphate, which is DTTP, there will be a lot of formation of this particular, there will be a lot of formation of this particular situation over here. And for this is that this over here is thymidine, the base, and that is complementary to the base guanine. So if there's a lot of DTTP, deoxythymidine triphosphate, there needs to be more of the complementary unit. And so this is explaining the substrate specificity.